Okay, good afternoon, everybody. Let me introduce you to Dr. Professor Jeff Johansen, who is a professor at the University of South Bohemia and mainly at John Carroll University in Ohio. And he will have talk for you about cyanobacteria, but because he's specialist on this group of organisms, and he's also interested in diatoms and other algae and ecology and taxonomy of them, and he will tell you more. Thank you.
Okay. I'm not used to a mic, but this will be good. So what I want to do today is talk a little bit about sort of the progress that's happened recently in the taxonomy of cyanobacteria. There's really been a real change over about the last 2 decades, of which I've been a part of. And so I thought this would be kind of nice to give you some stories about how things have really changed in cyanobacterial taxonomy. Okay. Yeah, so brief history. Cyanobacteria began to be studied around 1850. There were some real important early workers that started naming them at that time. And then there was a period where some critical reviews were published. These 2 papers by Gaumont in 1892 and Bournet and Flahal in 1886. They summarized what was known in the oscillatoriales and the Nostocales. And there's kind of this special provision in the International Code of Botanical Nomenclature where they can set different starting points in Linnaeus, and these 2 are different starting points. So these are the starting points for those 2 groups. The coccoid cyanobacteria go back to Linnaeus. Okay, so that was an important period. Those established the starting point, and then we had some monographers who studied cyanobacteria who sort of summarized a lot of the literature and did some of their own work in the mid-19th century. The most important of these was Lothar Geitler in Austria. He came up with a great big thick book that is in Dr. Bohunitzka's office and mine. And then there were several others. One, T.V. Dedzikacheri in India, Elinkin in Russia, Starmach in Poland, and Prescott in the United States. But mostly they followed Geitler's system. Now, all of this work up until this period of time, through the monographers, had some things in common. It was all based on microscopy and the light microscope. So, that was the basis. Sometimes ecology was considered important, but as time passed, people emphasized the ecology less. And it was actually kind of a bad thing because cyanobacteria are very ecologically specific, but people said if the morphology fits, they would use the names sort of casually or carelessly or whatever we want to think. There was also an idea that arose among microbiologists. It's called the Bass-Becking hypothesis, where the microbiologists said microbes are so small and so numerous that dispersal isn't a problem. So they're everywhere in the world, like a single species is everywhere. The environment will select. And what this led to was people like Geitler said, if you have a European flora, it applies to the whole world. Now, if you're a botanist, you know that the world has different floras on different continents, but they thought that these cyanobacteria were same species everywhere. And this really dampened taxonomic progress because when Geitler came out, he says, we're done. Here's the taxa. Okay. There was some more blockage to progress in taxonomy, and one of the chief problems was an American, Francis Druitt. He thought cyanobacteria were very plastic. He collapsed all the cyanobacteria, and there were about 1,300 species at that time, down to 125 species in 64 genera. Okay, based on the idea that they were very plastic in their morphology. Some ecologists liked it because you could name anything with this system. They all got the species. But another result of this was nobody wanted to name new taxa. If they've all been named and then collapsed, it's kind of like that kind of kills cyanobacterial taxonomy. Then another thing happened that killed taxonomy, and that was the work of Rosie Ripke and a number of microbiologists that were in her school of thought. She did very careful physiological work, and she sort of divided the texts up differently than they were in Geitler into different groups based upon, she looked at thylakoids and cell wall structure, kind of a microbiological approach with strains. But the problem was she was not a taxonomist, nor were the microbiologists that were following her. She did not describe species. She did not revise genera. She just used the morphology and promoted using the bacteriological coded nomenclature, which is very different than the botanical code where they've all been described, because it requires azenic strains. And it's very hard to have a cyanobacteria and get all of the heterotrophic bacteria out of the culture. That's just really difficult because they have these thick mucilage sheaths that the bacteria just love to live in. Okay, but they promoted this, and that also discouraged taxonomy because this is a very difficult thing to do. So we get to a period of time when there started to be a change in this. And Jiří Komárek and Konstantin Anagnostides were the ones that sort of broke the ice. They published a revision of the genera and families in 1988 to 1992. They came out in a series of papers. They took the finding of Rosie Ripke's work and revised the cyanobacteria. They said, well, okay, let's revise it. And they published 3 modern book-level reviews of the cyanobacteria, and these are the state-of-the-art reviews that we use to identify taxa today. Komárek described a lot of new species. He worked in Cuba and in Belize and had some very important floristic works and floristic works elsewhere. And Jiří Komárek is still active at an age of over 90 years. He's at the University of South Bohemia, and he is an amazing force. The cyanobacteriologists in Czech Republic, most of them came through his lab. And one thing I thought was really interesting about Komárek was he accepted change. So even though he's quite old, he adopted molecular methods. He says, okay, he doesn't do his own phylogenies or do his own molecular work, but he likes to include it in his work, and he is able to be nimble in his brain so that he could change from morphology to this kind of system. Okay, so now we're getting to a period of time where I'm going to talk about what was going on in my lab. And so I'm going to tell a few stories that kind of illustrate what happened. And I want to introduce this person. Sarah Boyer did her master's thesis at the University of California in Berkeley. Very well trained. She came to John Carroll as a technician on a grant I had to study soil crust organisms. And she's the one that established initial molecular protocols. I did not have training in molecular biology. She came and she started the sequencing, okay? She was actually going to leave academia, but she liked the small college environment. John Carroll's a very small college. And she thought, well, I could teach at a place like this. So she got her PhD at Harvard. Very good pedigree. She's now a tenured professor at Macalester College, and she studies the molecular systematics of spiders. But she was very important in my lab. And then another woman that was very important in my lab was Val Fleckner. She studied Chlamydomonas genetics, but she was denied tenure. This is the thing where you get to stay for a long time at a university. She won tenure on appeal, and then she came to see me because she says, this Chlamydomonas genetics is just too hard to do at a small university. There's too much competition. So would you like someone to work with? And I said, I certainly would. She learned green algal taxonomy and some cyanobacteria, and she was a microbiologist, great at isolating strains. We still have a number of her strains in our culture collection. Now, this is where it gets interesting. One day she called me into the microscope, and she says, I'm seeing this thing, and I have no idea what this is. And I looked at it, and I said, I've never seen anything like this before either. It's got spiral coiling, heterocytes, and false branching. So if you've looked at cyanobacteria, that's you have that. And there's nothing that was like that. So in 10 minutes, I said, that's a new genus. but we had problems publishing it. Okay, we ended up calling it Spiroressus rafaelensis, and these are a few more pictures. You can really see the coiling was really elegant in this taxa. It's a really beautiful cyanobacteria with a lot of morphology that a lot of them don't have. We first tried publishing it in the journal Phycology, and the reviewers demanded more information on the strain. They wanted molecular sequence data. We gave them more images, ecological data, and justification of the new genus, but not molecular data because I wasn't molecular at that point. I hadn't done anything. We sent that in. It was rejected again. We sent it in, and we actually appealed to the editorial board of the Journal of Phycology, and the editorial board, which consisted of a variety of people studying algae, said, you can't require molecular data if there's no molecular data to compare it with. So they were asking us to collect this molecular data, which is difficult, at that time, much more difficult than it is today. And yet it would be useless in a comparison because there was nothing to compare it to at that, 20 years ago. But the editor liked what the reviewers had said. They were microbiological reviewers. So she had our second revision. When she met with the editorial board, they made their decision. And she said that she had some reviews, but they were too informal, and she refused to give them to us. We think it's because they said that it needs molecular data. And so she got these reviews, which rejected the paper, listing a need for cell wall chemistry, fatty acid analysis, TEM, GC ratio, pigmentation, but not molecular data. So essentially everything that you use to study microbes, heterotrophic bacteria, except the molecular data. And so we said, okay, we give up. We'll give you your molecular data. And so we asked them, because at this time we didn't know what genes to use because nobody had been using them. We said we could use 16S, which was possible to get, or we could use the internal transcribed spacer. And what do you think we should do? And we got a response back from the associate editor, well, you need to do both, and you have to sequence all the comparison taxa. And we just said, the hell with the Journal of Phycology. That was too much. But we did sequence the 16S and the ITS because Sarah knew how to do that. And we sequenced some comparator taxa, these genera here, Tolipothrix, Calothrix, and Coleodesmium. We submitted it to Nova Hedvigia. Again, there was a difficult review process, but they accepted the revision, and it was published in 2002, 5 years after we first saw it in the microscope. So this project took a long time to publish, but it does represent actually the first validly described and named new cyanobacterial genus using what we call the polyphasic approach. It uses molecular data, morphological data, ecological data, and whatever data you can get your hands on. It was actually well received by a number of cyanobacterial researchers, and I'll just give you an idea of what some of the things we did in this paper. I look at it and I consider it sort of primitive after 20 years of doing taxonomy. We compared the morphology in 2 different media. We had 2 different strains. We did that kind of work. So we had the morphology. We had a classical description. At that time, if you wanted to describe an alga, or any plant for that matter, under the code, you had to have a Latin description. So we have a Latin name, a Latin, what's actually called a diagnosis, is very old-fashioned. There's the Latin above, but it translates, differs from Cyanidemia and Tolipothrix in that the filaments are regularly, tightly coiled and right-handed helix. So that's a diagnosis. You just say it's just a comparison with the close taxa, and then you have a description, okay? We also had to have, we had to set aside a type, and you do all of this stuff. And then the etymology is how you got the name. And Spiroressus means spiral rope. And I don't know how much background you've had with cyanobacteria, but there's a genus called Spirulina, which means spiral thread. So this is a big, thick thing versus the thin thing, the Spirulina. Okay. We use primers that will amplify the, this is a diagram of the ribosomal operon, and it consists of this 16S, which is also called the small subunit. And then you can see where the ITS is. I can point that out, I guess, with this, yeah. So here's the ITS. And the ITS can have 2 transfer RNAs inserted. And then you get the 23S ribosome. That's actually much bigger than the 16S, and there's a 5S way out there, okay? But we had to have these primers that would amplify, and at this point in the game, we were doing one amplification from 2 to 6, and another amplification from 5 to 1, because we couldn't get the whole thing in one sequence at that time. It wasn't possible. This is a very sparse phylogeny. You can see we just have Spiroressus, Tolipothrix, Colliodesmon, Calothrix, one strain of each, and this Cytonema. Okay, you know. So we were cloning. That means you take the PCR amplicon and you put it in a clone, and that way you can see the ITS. You can't see it otherwise because there's multiple operons and it doesn't give you very good results. Kind of an interesting side note, this right here, we had these really huge differences between the clones. We didn't know what it meant, but it meant that there were these radically different operons, and what was it? It was 15 years later, we published a paper on that topic. Dr. Bohunitska is on that paper, and collected a lot of the data for other cytonemas in this group. So that was kind of fun, but it waited a long time before it happened. This is also a very crude analysis. This is just a diagram showing the length of the different ITS regions and whether or not they have transfer RNA genes. So that's out of the paper. And you know, we looked at the genetic similarity. So generally, they thought at that time that you know, if you were less than 97.5% similar in your 16S, that meant you were a different species. Okay, Spiroressus is over that. Both Tolipothrix and Colliodesmon look like they would be the same same genus at least, but maybe even the same species. This value is really high. Okay. But the similarity threshold indicates different species, but if you're above that, it's considered meaningless. So we went ahead and named them. There were no genetic thresholds for genera at that time. Okay. And then this is one of the concluding paragraphs where I cited Komarik's work. So Komarik said they recommended recognition of small, unambiguously defined genera, and said they indicate no particular purpose is served by maintaining large genera and subdividing them into subgenera, sections, subsections, etc. And we said, given the genetic and morphological similarity of Spiroressus, Tolipothrix, and Colliodesmium, seems our choices would be to expand Tolipothrix to include them all, or to define these smaller genera. And we preferred the latter choice. Now, I say that because that was Komarik's idea, but this was the way we introduced this into the molecular data, and that's become sort of the standard in the field. That's what I think everybody follows, this idea that you need these smaller genera that are better defined. Spiroressus was actually cited, this paper cited in 135 papers. Nova Hedvikia has almost no impact factor, but people still grabbed it and looked at it. And so, because it is the first one where we had this thing called the polyphasic approach. Now I'm going to skip through something. So we had 12 years after that where we described 5 genera and 30 species. I worked with lots of Czech scientists, including Marketa. And during that time, we improved our analysis. We got better sequencing technology. Now we get much cleaner sequence, and we can actually do a single read that's all long, and that's much nicer. We have better phylogenetic methods, and I don't know if you've had any of these, but when we started the work, it was parsimony was used. That's not nearly as good a criterion as maximum likelihood, which replaced parsimony, and now we use Bayesian inference mostly because that's even better method. So we have better phylogenetic methods, and we had better analysis of the ITS regions. So this is a time when we went from those bars that said how long they were to the secondary structures. And so I'll show you what this looks like. This is a secondary structure of the whole ribosomal operon. So what happens when you synthesize RNA is you have your region up here called a promoter, and the ribonuclease comes down and it starts making RNA. And so this long piece here is what we call the leader. And then we get to this gravy thing. This is the whole 16S. I just put it in a little triangle, okay? Everything between this triangle and that triangle is the 16S to 23S ITS region, okay? And you can actually see that there's another 5S, and it actually, this 23S5S binds with this thing, and the leader binds with it, even though this is like 1,500 bases, and this is, I forget exactly what that is. I think it's over 2,000. But we have these structures in here that form. The number one is the D1 prime helix, and then we also have this box B and the V3, and those are the three that are most important in taxonomy. We use those. If you have both transfer RNAs, then you usually have a V2 in between it. V stands for variable helix. And this is what the D1 prime helix looks like. In a comparison of a paper that was done fairly recently, what you can see is these are actually called Fulbrightiellas, and you can see they look similar, but there are differences. There are differences in how they open up in the helix, okay, because of mismatches. This is Sherwoodiella, another genus that we were describing. You can see it's quite different than this. And this actually we had two different operons with both tRNAs and no tRNAs. And then this is a Calothrix, really looks different, okay? So that's how we use. So that was something that happened over that 12-year period. And the next big advance in our taxonomy work, I thought, was starting to name something called cryptic species, okay? Now, this is a fairly recent thing that happened. It's kind of like with the molecular data. It's happened in a lot of different taxa. They've been named in many animal, plant, and algal groups. And I've just got a few examples here, like the Anopheles mosquito. I think there's 7 different species that you can't tell them apart morphologically, but molecularly you can tell them apart. Here's a fish that they look pretty similar, but they actually are genetically quite different. So those are different species. And even this is a small alga called Cenodesmus. Even with scanning electron micrographs, there's some of them that look identical, but they're different in terms of their genetics and have been named different names. But they had not been named for cyanobacteria. And so the first paper to do that was in Oculotella. So we had these Oculotella species where we named cryptic species. And now I would just say people are naming cryptic species all the time. But this was kind of like a new idea, and a lot of people didn't like it. They don't like the idea of naming something that's so cryptic you can't tell what it is without getting sequence. Oculotella subterranea, that's the type species. It was described by Gabriel Zammit in 2012. After many years of study, this was actually first isolated from Roman catacombs, and by Patrizia Albertano. Zammit was her student, but Patrizia was publishing on this but not naming it. And it has these distinctive, sorry, hit the forward, has these distinctive dots, structures at the ends of the trichomes. It's probably better seen here. You can see there are little reddish dots, okay? Now, those are actually rhodopsin, and this is a molecule that is light sensitive. These actually have what we would call an eye spot at the tips of the filaments. And it's the only cyanobacteria with an eye spot. So all of them have it, and anything outside of it doesn't. So it's really easy to identify the genus because of this. So while Patricia had hers from catacombs, we'd been isolating a whole bunch, and we had a whole bunch of these. And so when she named the genus, we were ready to go with a paper on it. These are the different ones that we have. You can see they look a lot alike. But what I wanted to do was say, okay, well, in justifying these cryptic species, we should say they occupy different niches. That's one of the things about niches is that we say no two species can occupy the same niche, okay? And so is there niche separation? So the first place, Subterranea, was found in Hypogea of Italy, Malta, and Israel, in these dim light, humid air environments, no flowing water. Lights have been put in because people it's a tourist thing. You go into the Roman catacombs and there's art on the walls, and the algae was growing on the walls and ruining the art. So that's why they invited the phycologists in to look at them. Now, these habitats have a very limited diatom flora, and what I'm using is diatoms are very specific, and they could tell us if the niches are the same. Okay, and this had these diatoms called Ludicola and Humidiphila, and that's where we got this oculotella. Another site that we collected was this is Calf Creek Falls, and Marquetta was there when we collected this, and the diatom flora there had Cimbella and Cyanopsis, Gomphoneum, and Navicula. Completely different than the subterranean habitat in the catacombs. And Oculotella cataractarum was described from here. And then in Hawaii, we collected some stuff in this sea cave, which had a diatom flora dominated by Diprora and Itzia, very strange diatom flora. So Oculotella kawaiiensis was described. Fero Hindak, a prominent Slovak scientist, isolated this oculotella from a eutrophic lake in Austria, the Hafnersee. So Oculotella hafneriensis was described from there. And then Lubasz Kovacic, also a Slovak scientist, he would go on vacation, he would collect some stuff from the place he was visiting, like in Greece here, Nea Kameni Island. And Oculotella neokameniensis was described from there. He's on that paper too. We have these sites in the arid deserts of North America, in the Mojave Desert. They're actually very similar habitats, but molecularly they were different. And then in the Atacama Desert, in the coastal fog area in Chile, we found another Oculotella, and we called it Atacamensis. So the first four species were in very different habitats. The catacombs, waterfall, Hawaiian cave, and lake. You could tell those were apart. But then the last four were all in arid soils. And so those are kind of a similar habitat, might be the same niche. So we looked at morphological separation. These are the line drawings of these, and these are to scale. There were some minor differences, but really very cryptic, very cryptic. You'd have a hard time telling them apart. We did measurements, and I'll just point out, like in this trichome, the trichomes were 1 to 2 microns wide, which is not very much of a variation. We actually did some statistics. They were statistically different, but there was so much overlap. These were really cryptic. And then this is our phylogeny. you'll notice it's much more full than that Spirostis phylogeny, because now people were sequencing and we had lots of sequences and lots of things we could compare it to, but the Oculotella was all up there, and this is the tree showing just the Oculotella, and the taxa were quite distinct. Every species was monophyletic in its own clade, and so we had good separation phylogenetically. You can see the D1, D1 primes. Some of these are really similar. The group is very similar. It's the soil group plus Hafneriensis, and then this is another group that they're even more different, Cataractarum subterranea and Kauaiensis. This is not Oculotella, it's just there for comparison. And the V3 helix was what was really different. So that gave us a lot of things. Now, one of the things I like about secondary structure is it's almost a little bit like morphology because it's visual. You can look at it and you can say, oh, that looks different. So that's kind of a nice way of using some molecular data. And right at this time, the thing that really made some progress with cryptic species was a pair of researchers, Irwin and Thacker, had been looking at Senecacoccus that lives in sponges. So it was endosymbiotic. They couldn't grow them in culture. They were microbiologists. They says, well, we can't grow these, so we can't identify them, but we can tell they're different species. And what they said was, within species, the p distance, so it's kind of like how percent different kind of thing, but the p distance was 0.83 between species, within species, but between species, it was 9.32. So there's a huge gap, a huge thing separating whether you're in the same species or you're in different species. And we applied this to Oculotella, and we got almost the same numbers. And so it really worked very well. And so this is one of the criteria we used, and we still are using this criteria to identify cryptic species. So, did we have sufficient evidence to actually describe these things? So I'll just give a summary of the evidence. All our strains were very distinct from the type species. So Oculotella subterranea was red. Easy to tell apart, okay? Cataractarum and Kauaiensis are morphologically, ecologically, and molecularly and biogeographically distinct also. They're different from everything. Hafneriensis is from a lake, quite different ecologically, but similar morphologically. And then we had these three soil species, Neocominiensis, Mohaviensis, and Colburnii, that really were very, very similar with minor differences. They're very cryptic. And Atacamensis is also in this group. These are, by the way, the wider ones. The ones that are up around 2 microns wide. So, and then there's a species concept, phylogenetic species concept, where they say, you should be identifying anything worthy of taxonomic recognition. So we asked that question, and we said, the evidence is good that they are genospecies, another word for cryptic species, and separation was possible using this polyphasic or total evidence approach. And morphologically, there was a statistical difference, but they were still pretty cryptic. We considered them worthy of taxonomic recognition because they were in such different places, and we were thinking, if you didn't name these, nobody would be interested in them again. Okay, they just call them Oculotella subterranea, and maybe one of the wider ones, and that would be it. So we were happy that we did that, and now people are doing lots of cryptic species. So the question is, where are we at now? There's a real problem with multiple operons. So the ribosome, you get several ribosomal operons in some taxa. Some genera only have one, but most have two, and you can have up to 5. That we've seen that many. And really, you should only compare the operons that are the same. So these operons are recognizable if you have enough data. And so you should be using orthologous operons, not paralogous operons. You shouldn't mix and match these because they're going to be very different. And if you're using these thresholds, it'll throw it all off if you're using the wrong ones, okay? So we're working on ways to try to recognize different orthologs. And there's a paper going to be coming out in FATEA, which is the journal of the Czech Phycological Society. And again, Dr. Bohuniska is the lead author on that one, on Brasilanema. And we really had to struggle with this because we had lots and lots and lots of Brasilanemas that she'd sequenced, but we didn't have every operon for every taxon. So it really was one of these things where we had to figure out how do you use this complicated data set to do things. So that is one paper that did it. And then I have a paper in review right now on a genus called Dulcicalothrix, where we actually look at the indels. So this D1 prime helix seems to be the thing that helps us identify operons. And both in Brasilanema and Dulcicalothrix, I did that. An indel means you have a gap. There's a bunch of nucleotides missing in the sequence. So that's an indel. And these ITS often have indels. And now what's really kind of interesting, we're also returning to Spirorestis. So after 22 years, I'm working on Spirorestis. It turns out it's a large clade of soil strains. So you can see up here, that Spirorestis, there's 43 sequences up there. All of these are either aquatic or these half circles are subaerial, kind of on rock. There's only 2 soil forms in this whole area down here. But then when we look at the ITS tree, we can essentially see 5 new species. 5 new species of Spirorestis, 2 new combinations, meaning we're going to take them from the genus Thessalia, where there are now, and put them into Spirorestis. And then, of course, there's still Raphaelensis. Now, what we found is the coiling isn't consistent. So a lot of these are not coiled at all. And that's how we recognize Spirorestis. But what really turns out is the habitat is consistent. These are all from soils, mostly from desert soils. And so that is actually more important than the morphology. So the ecology is more significant. And this is just one of the ones, this is the one from India. I have a colleague there who said, I'd like to describe this, and that's what got me back to Spirorestis. So now I actually think that cyanobacterial taxonomy, the work is changing. We're entering a period, I think, where we're going to start doing more revision. I think there's been genera described that maybe they went too far, or species where they shouldn't have been, they weren't aware of the literature and they described things that had already been described. So I think that's going to happen. And I also think that a thing that's going to happen is we're going to describe species and put them into existing genera. There's been this trend of describe a genus with one species from one strain. And now we're finding, when you isolate enough of them, we're finding these things. So for example, Spirorestis was for a long time only one species. Now it's going to have like 6 species. And that's happening in a lot of these. We're filling out the taxonomy. And I think that's the period of cyanobacterial taxonomy we're in now. So, a few final thoughts. Cyanobacteria are now being studied in many labs in different countries and on different continents. So this is actually a really, I say almost a hot area. There's lots of things being published. Czech Republic and Germany are critical, okay, in that effort, the labs there. USA and Mexico, Brazil, China and Thailand, Russia and Ukraine, and India all have significant labs doing significant work on cyanobacterial taxonomy. The methods developed by my research group are being used throughout these studies, so it's actually very exciting to see others adopt these methods. There's been more than 60 new genera described in cyanobacteria since 2000, with many more species. And so, we went from a period where there was nothing being described to a period where it's become very active. And many students have been trained in cyanobacterial taxonomy and have gone on to PhD programs, and many of us established their own labs from my lab. So, for example, Dale Casamata was a very important early person in the lab. He's in North America. He's an American. Nicole Petrosiak, a German, was also really important in my lab, and now is doing wonderful work in University of Nevada at Las Vegas. And then I'll just point out we've got Marketa Bohanitska, Radka Milsteinova, and Klara Zhirhakova, all out of my lab. So Czech scientists are still at the top. So, and I think it's really good. And that many Czech scientists didn't come to my lab. So, but this is the group that came through it. So this work is really exciting because it's discovery science. We go out and we discover new things, and that's kind of exciting. It's exciting to describe new genera and describe new species. You're seeing something for the first time. It's driven by evolutionary hypotheses, so we have these evolutionary species concepts, and we're looking at the evolution of these organisms, so that's also really interesting. And this work is received by good journals, so you might learn that you want to be publishing in journals that have impact factors and these kinds of things going on with them. So you can do that with this group. And I hope I am still publishing when I'm 90 years old. Professor Kamark is a real example for me. He is an amazing person. If I were a Czech, I would be he would be part of my pride in the country. He's that kind of a stellar light. He's published over 400 papers in green algae and cyanobacteria. So he's been a leader all his life and is still an amazing scientist. And thank you for listening.